Hi, everybody. So today we have the pleasure to have Dr. Fairchild. Tim completed his undergraduate and postgraduate training at IIID, where he focused on the metabolism of carbohydrates. Prior to commencing in his current position at Murdoch University, he spent 3.5 years as an assistant professor in the Department of Exercise Science at Syracuse University in America. Tim's area of research revolves around the management of obesity and obesity-related disease and conditions. Tim is particularly interested in the interaction between diet and exercise, which now forms much of his research focus. So today, the title of his presentation is Management of Blood Glucose and Exercise, the why, how, and when.
Thank you for the introduction and thanks for the invite. I'll pretend I had nothing to do with that little bi-sketch introduction. But basically I will talk, I'll be presenting, I guess, some of the work that we've done previously and then some of the work that we're currently involved in. I am based in the School of Psychology and Exercise Science. So, you know, we are hoping to make a few more kind of links in and around the university with different researchers. So I guess the question is, why exercise? So exercise is unique in its ability to transiently upregulate a lot of metabolic pathways, and it really challenges the system to a degree that's not normally seen in healthful sort of conditions. So as an example, we've just finished a little project where we looked at the role of exercise on the metabolome. And if you can see on the left, we've got all the different metabolites that we're able to pick up. And basically what we're interested in doing is just identifying the metabolites that changed. And you see a broad change in a metabolome. Now, please remember, this is plasma, so it's not within the muscle. So you'd expect greater changes, you know, within the cell. And, you know, we're up to have baseline written in here. In this sort of an exercise challenge, people sat on the bike. We did what's called a time to exhaustion challenge, where people sat on the bike at a certain workload and stayed on the bike until they could no longer pedal. So the baseline was actually 15 minutes into exercise. So if we used the resting condition, we would have seen an even greater sort of shift in the metabolome. So even within exercise, so from a 15-minute time point to roughly 45 minutes into exercise, we still see quite large changes. And if we kind of map the particular cluster, so in here, through the work with Rob Trengove and David up at ECU, we're able to identify particular metabolites that have very similar trajectories. We're able to see significant differences between the 15-minute time point to pre-fatigue, post-fatigue, and 20 minutes past fatigue. And the reason why this is of interest, not only to sport, but obviously in clinical conditions, is we can take healthy individuals, we can transiently fatigue them, identify specific metabolites that are involved in this, and then the metabolites that come back quickly post-fatigue to see whether or not there are particular markers that herald the onset of fatigue and whether we may see this in clinical populations. We've only just finished this study. It has been submitted, but you know, this is basically, you know, one area of, I guess, research that we go into. But the reason I present this is not to talk about the metabolome, but rather to talk about the fact that exercise changes many things. We see huge changes in metabolism, but also we see large changes in, you know, a whole heap of markers. So myokines were first, I guess, coined the term, I believe, in 2005, 2006. And really, we know now that the muscles, you know, goes beyond simply contracting, but actually releases a whole host of cytokines, as well as small peptides such as irisin, BDNF, and things that have, you know, influences in different parts of the body, and FGF21 in particular, which is an area of research focus for us with appetite and things like that. But we see huge changes within a lot of these peptides that are released, you know, in and around the body, and that have quite clinically meaningful changes. So exercise, this is why we're interested in exercise. So I guess why diabetes? Well, diabetes globally is a leading cause of blindness, kidney failure, and amputations. It is a leading cause of mortality in and around the world, and this trend seems to be worsening. You know, beyond mortality, every year, 0.7% of adults develop diabetes. Now, this is fairly old sort of data based, I think, from about the 2004-2005 AusDiab study. But again, we wouldn't expect this to have changed much in the last 10 years or so, and if anything, may have gone up slightly. But diabetes is a major concern in and around the world. What is the relationship between diabetes and exercise? Well, when you look at the epidemiological data, so here what we've got is individuals who are inactive and their risk, the risk of new incidence of type 2 diabetes. So these inactive individuals conduct 0 minutes of exercise per week. Insufficiently active is 1 to 149 minutes per week. You know, we often talk about 5 bouts of 30 minutes of exercise. Sufficiently active is 150 minutes per week. I do want to emphasise this is sufficiently active to maintain health, not to improve health, but to maintain health. So if you walk 30 minutes 5 times a week, you are in this sort of a category. We're not talking athletes here. You know, so we are talking simply doing 150 minutes of just, you know, low to moderate intensity exercise. So when you look at the increased risk, we see, you know, as we become, decreased risk with an increased level of activity or physical activity. And, you know, this is mirrored in, you know, epidemiological studies around the world. Okay, so how does exercise have this effect? Well, you know, most of us will be fairly familiar with how glucose is regulated. So plasma glucose circulates around the blood and it translocates, or it comes within into the cell via particular types of transporters called glucose transporters. GLUT1, first transporter to be discovered, is ubiquitously expressed, and under resting condition is the main one that glucose utilises to enter a cell. When we have a large increase in glucose, it spikes the release of insulin from the pancreas, which translocates this intracellular pool of glucose transporters, GLUT4, to the cell membrane, and we see a large increase in glucose uptake. And the increase can be anywhere between 10 to 40-fold. It is fibre type dependent, so we do see differences in type 1 versus type 2 fibres, which again hasn't garnered a lot of attention. But there are differences within particular types of muscle fibre types. And these GLUT4 transporters are not only located on striatal muscle, but also adipose tissue. And, you know, I don't want to go into too much, but if you go through an overfeeding model where you increase glucose uptake, often what you'll find is a resistance within the muscle, but the sensitivity in adipose tissue may actually not change and can sometimes increase. Okay, so, you know, when we talk about insulin resistance, please bear in mind, you know, often the idea is that it's a systemic insulin resistance, but the reality is there may actually be some adipose tissue, visceral adipose tissue, for example, that may actually not become resistant to the actions of insulin. So, you know, just be a little mindful that, you know, again, that hasn't really been deeply explored. Most of the studies are done on clamp studies, which are fantastic, but really when you look at tissue-specific changes, most of the work's been done in animals, and the work back in the 90s seems to indicate adipose tissue doesn't seem to get the same resistance that we see in muscle. However, muscle predominates, so we have a large amount of muscle, so hence it is, you know, it takes care of most of the glucose within the body. And please remember, in the blood we only have 4 grams of sugar or 4 grams of glucose, so it's not a huge amount of glucose that actually circulates in the blood. Okay, so insulin is released. How does insulin do this? Well, you know, this is the basic signalling pathway with a few steps removed, but insulin basically is one of the main triggers for shifting the GLUT4s towards the cell membrane. We talk about insulin sensitivity, and I used that term a second ago. So insulin sensitivity and its inverse is insulin resistance. So healthy individuals are typically individuals who are normal glucose tolerant, are typically what we consider insulin sensitive. So the more sensitive you are, you know, you have a greater glucose uptake per unit of insulin that's been released. Insulin resistance means you need a greater amount of insulin to take care of the glucose that's circulating. Insulin responsiveness is slightly different to insulin sensitivity. So one other thing that we do see is, in particular, exercise not only, and we'll see this in a second, not only stimulates the translocation of GLUT4 to the membrane, but it also switches on, basically switches on the pathways to increase the GLUT4 protein. So within about 18 hours, we see a greater amount of GLUT4 within the muscle, so hence we have a greater responsiveness. So our maximal capacity in the cell can actually increase. So insulin responsiveness is the maximum glucose transport capacity within the cell, and this can vary and fluctuate. The sensitivity is basically at low levels, how sensitive is the muscle to glucose. We know that when the system fails, it leads to insulin resistance, and typically the progression is towards, or often the progression is towards type 2 diabetes. Okay, so as I mentioned, basal condition, basal conditions, we see glucose transport, and these are just arbitrary units. We see a certain amount of glucose transport. When we add insulin to the system, we see an increase in glucose transport, and that's because these GLUT4 transporters or transporters have been translocated. They're now based on the membrane, so more doors are open for glucose to come in. We also see a similar effect with contractions. Okay, so if we take mouse or if we take muscle, we contract the muscle, we add caffeine to muscle, we see an increased translocation of the GLUT4 to the cell membrane, and we see an increase in glucose transport. And these two are somewhat additive, not completely additive, but they are additive. And John Holossi was a person who really did all the bulk of the work in the late 80s. That basically focused on this contraction and insulin sort of system. So what this seems to suggest is that there are two different signaling pathways, and indeed, a lot of work that's now done is focusing on which pathway is the most important pathway involved with exercise, and different groups believe there's different pathways have specific roles, but calcium is probably the most likely pathway involved in this, and the reason why this is clinically relevant is the fact that I'll come back to that in a second, is that when this fails in type 2 diabetes, this pathway is still working. Okay, so in individuals who are insulin resistant, where the binding of insulin really doesn't translocate these GLUT4s, exercise and contraction still does. So this is really where a lot of the work and why a lot of that work is continuing. This glucose transport does reverse within the first 2 to 3 hours, and hence this is why athletes are typically told to consume their carbohydrates within the first few hours after exercise, because with exercise we get this translocation of these GLUT4s, muscles are open for business, you take whatever glucose supplement it may be, that glucose can now enter the muscle and hopefully replenish the glycogen. So this translocation, these GLUT4s typically sit there for a couple of hours. The insulin sensitivity does persist for up to 18 hours, but again, this does depend on what's being consumed. Okay, so in different situations, if you have somebody who exercises and then he's given a high-fat, very low-carb diet, the insulin sensitivity seems to maintain for a lot longer than if carbohydrates are replenished very rapidly. So the sensitivity does seem to be influenced by the types of foods you do consume immediately after exercise. Okay, so that's basically the why. So this is why we've been interested in exercise. Changes a lot of things within our biochemistry. Type 2 diabetes is a major concern, and we know that exercise seems to improve the glucose management, even if type 2 diabetes has been confirmed. So we did a study, this is a few years back when I was still at Syracuse, one of our PhD students there, and what we're really interested in doing is looking at, okay, well, we'll look at the effect of sitting around in a lab all day from 7 a.m. until 7 p.m. while we sample blood every 10 minutes. So people came in, sedentary condition, literally sitting in a chair. If they need to go to the bathroom, they were wheeled out and basically taken to the bathroom. And in this study, there was actually a third condition where rather than exercising for 60 minutes first thing in the morning, we also had them do 12 bouts of 5 minutes every hour. So it was this kind of, breaking up the sedentary period. But really the main focus of the talk today, I'll only present the data on the 60 minutes. And basically this is the sedentary. So they were provided meals, 6 different meals, around 300 or 400 calories from memory. And obviously this is blood glucose concentration. So what happened when we exercised them first thing in the morning? Okay, so this is a 60-minute exercise bout, at a moderate intensity. What does that mean? Walking relatively quick. You can maintain a conversation, but the conversation will be, kind of staggered a little bit. What happened to blood glucose? To be honest, not much at all. Okay, so if you have a look at this, this is when they were performing the exercise at the first part here. Blood glucose was a little bit lower, but if you look at the remainder of the day, okay, blood glucose seemed to be a little bit elevated for the remainder of the day. And again, this is in the lab, we were providing the meals, so we were a little bit surprised at this. That intermittent exercise seemed to be a bit more effective in managing that diurnal pattern, and hence, this kind of big push to break sedentary activity patterns. But really, we were quite surprised because, look, 60 minutes, it wasn't hard exercise, but at the end of the day, it was, people still performing some exercise. To give you an idea of what the difference is, because it can be difficult to see, all we did here is we just took the values that you see at this first time point and subtracted the sedentary. So for each individual, we took their exercise day first time point, subtracted the sedentary time point, and then time point 2, time point 3, time point 4, just to see, how it actually varied throughout the day. So if they were exactly the same, we would expect it to go straight along this zero line, okay? But basically what we were finding is that the exercise was effective in bringing the values down slightly while the exercise was being performed, but you can really see, glucose kind of sat above the line for the remainder of the day. And there are really no statistical differences between, doing no exercise and doing the 60 minutes of exercise in a day. This is insulin. We looked at insulin, C-peptide and glucose because we were quite interested in the pulsatility, the release of insulin, hence the rapid or the frequent sampling. And really, when we looked at some of the dynamics or the kinetics of insulin being released from the pancreas, it didn't seem to have a large effect or a large difference between exercise or sedentary sitting. Insulin seems to be a little bit more responsive. Okay, so we exercised. If you can see this, insulin seems to be suppressed to a greater degree during exercise, which is expected. When we exercise, the release of epinephrine or the catecholamines suppresses the release of insulin from the pancreas. And if you look, most of the day kind of sat below the line, which again is not that surprising because we would have thought some of those GLUT4 transporters would have been translocated to the cell membrane. So glucose can come in. There was really no need for, insulin to be released from the pancreas because the GLUT4 translocation had already happened. But, like I said, I'm kind of making a little bit of a bigger deal out of this than it was because there weren't really large differences. So why didn't we see a bigger change? Well, the reason why we probably didn't see a bigger change is we've got to separate local and systemic effects. Okay, so locally, the muscle probably was increasing glucose clearance. Okay, so the muscles that were exercised were probably taking glucose up at a greater degree. But these are just the contracted muscle. So systemically, we didn't really see a big difference. Okay, so I'll take you, this is not our work, so this is from a different group, a Scandinavian group. And they did a really neat study where, they used a clamp and exercised and then provided a meal afterwards. So, one of the things I just wanted to highlight, so 3 different conditions. They took overweight and obese, normal glucose tolerant. So individuals that have no glucose impairment, overweight, obese, impaired glucose tolerance. So basically, an increased sort of glucose response to a glucose challenge. And then overweight, obese, frank type 2 diabetes. Okay, this is the plasma glucose. Resting condition, exercising condition. So start off with rest, then oral glucose challenge, which is 75 grams of glucose, and then just measured the glucose shifts. Okay, and one of the things that you'll see is that, well, really, when you look at plasma glucose, it, similar to what we found, it really didn't have much of an effect. And in fact, if anything, the exercise was somewhat negative. So performing the exercise, having the glucose challenge immediately after the exercise seemed to actually increase the glucose response. In overweight, obese, and type 2 diabetics, it was basically, exact same sort of responses. Well, what about this kind of, first of all, I wanted to talk about the rate of disappearance. And apologies, the animations, I'm probably a little bit behind. I think I finished this presentation at about 10:45 this morning. But if you look at the rate of disappearance, so like I mentioned, they're very interested in looking at where the glucose, so how quickly the glucose is entering the system and how quickly it's leaving it. And they were able to do this because they were doing what's called a clamp. And then, so in essence, they're infusing a type of glucose where they can measure how quickly the glucose is leaving. And then the drink that was ingested was labeled as well. Okay, the infusion was labeled with deuterated glucose, and then the drink was labeled with carbon-13. So they're able to tell whether or not the glucose was coming in from the drink that was ingested and how quickly the glucose was actually leaving. So it's quite a neat sort of study. So the rate of disappearance here. So if you have a look at the bottom section, you will see that the glucose during the exercising condition was leaving the plasma quicker. So in essence, we were seeing an increased rate of glucose disappearance, which means that the glucose was actually leaving the blood a lot quicker following the exercise versus the resting condition. However, the glucose was also appearing a lot quicker. And obviously the glucose was appearing at a faster rate than it was disappearing. So, and this is basically what I mean. So if we focused only on the muscle, we would have seen exactly the same thing that we would have expected, which is that glucose is entering the muscle nice and quickly, but systemically, glucose is also appearing, okay? And this is appearing from somewhere, okay? So where is the glucose appearing from? Well, some of the glucose is appearing from the liver. So endogenous, so when we think about the liver looks after our glucose concentrations within the blood. Okay, so endogenous release of glucose was increased. Okay, and again, this makes sense. When we exercise, we release a whole host of hormones, in particular glucagon, which stimulates the release of glucose from the liver. So, and you can see this was increased during exercise. So the liver started kicking glucose out, and at the end of glucose, well, the liver's now releasing glucose. Okay, you've now ingested glucose, so all of a sudden you already have this increased release of hepatic glucose, plus you've got the glucose coming in from what you've just ingested. Okay, one of the other surprising findings was that the rate of appearance of the glucose drink increased. Okay, so why would we see this? Well, part of the reason why we see this is there's some intestinal disruption when we exercise. So when we exercise, there is some intestinal disruption, so glucose is likely to be just absorbed quicker. And most studies are finding this. The actual cause for it, they use the term intestinal disruption, but the reality is we don't really know what it is. Because in humans, it is all through the epithelial cell. So there's no between epithelial cell sort of translocation of glucose, to our knowledge, like what we see in other species such as chicken. But really, this is all transcellular. So, but typically what they're seeing is an increased rate of appearance of glucose from the drink. Okay, so exercise does increase glucose clearance, but the rate of appearance is also increasing. Okay, and this one other thing I just want to highlight, this effect, if you look at the liver, seems to kind of have this effect for around 60 minutes. Okay, and the reason why that's important you'll see in a minute. Okay, so we do need to be a bit careful because when we compare local versus systemic differences, locally we are seeing an increased glucose uptake, but systemically there are a lot of other things happening. With exercise, we upregulate or we increase the concentration of epinephrine, glucagon, cortisol, growth hormone. All of these hormones have somewhat of an insulin or impair the effect of insulin or stimulate the release of glucose from the liver. So they all have an effect on glucose regulation. The other thing that we do have is inflammation. Obviously, inflammation's been linked with insulin resistance. So when we exercise, a day or two later, we have pain in the muscles. Well, exercise is also associated with inflammatory markers. So we have a whole host of things happening systemically, which might be impacting why we're not getting this effect locally. Why we're not seeing this effect systemically. The other question is how much exercise contraction is needed to translocate GLUT4. Okay, so how effective or how strong does that stimulus need to be to actually cause the translocation of GLUT4? Okay, so we do know that high intensity exercise or high intensity stimulation causes a greater translocation of GLUT4 than low intensity. So just kind of walking around is probably unlikely to be a strong enough stimulus to cause the translocation of GLUT4. So the question really is, and it does remain unanswered, how intense does the exercise need to be to really speed up this kind of GLUT4 translocation so that we can increase glucose uptake into muscle? Okay, so one of the studies that we looked at, or the next study I'll talk about, is the effect of exercise type, performed an hour before an oral glucose tolerance test. Why an hour? Because one of the things you'll notice from that previous slide is that the liver kicks glucose out post-exercise for around about an hour. So we thought, okay, well, we'll initiate the meal about an hour afterwards and just have a look to see what the effect of a prior exercise bout would have if we conducted or if we kind of ingested 75 grams of glucose. The population were overweight, obese individuals, normal glucose tolerant, and two exercise conditions: continuous exercise for 30 minutes or HIIT exercise. We're not talking Michael Mosley HIIT, 2-minute sprint sort of stuff. We're talking hard exercise, easy, hard, easy, 6 repetitions of that sort of a cycle. We performed an oral glucose tolerance test the day prior to exercise. We then did exercise, then we did our oral glucose tolerance test, and then we had participants do an oral glucose tolerance test again the subsequent day. So we basically did an oral glucose tolerance test day 1, day 2, which was after exercise, day 3. 1 week they came in, they did continuous exercise before. The next time they came in and did high intensity intermittent exercise. Okay, so what did we find? Well, not a lot. But Aaron's writing his PhD up on this study at the moment. But basically 3% improvement, really not a big shift at all. And I've just presented the area under the curve. Large variance, small sample size, which may have accounted for the large variance, but we are seeing fairly large variance in some of these responses. On day 2, so this is post-exercise, there was a decrease in area under the curve. But again, be mindful, the y-axis is being cut here, so when you look at this difference, it really wasn't a huge difference. It was around 3%, 3.4%, I believe. The day after, oral glucose tolerance or the area under the curve was similar, and really there were no differences between either exercise condition. The other thing we did is we had a CGMS. So this is the continuous glucose monitor. So participants had a continuous glucose monitor placed. This measures interstitial glucose, and we get samples every 5 minutes over the course of the 3 days. Okay, so we looked at day 1, day 2, and day 3 to see, well, did that effect of exercise maybe kick in later, which we didn't really expect based on some of our previous lab work. And again, we really didn't see a lot. I've got to be honest though, we used the CGM units and the data coming out of these wasn't great. So when we kind of looked at some of the numbers, they did seem a little off. And a lot of these units, with those AccuCheck sort of units, glucose units, they need to be within 20%, 80% of the time. Okay, so when you look at those, it's a fairly large variance when you look at some of those units. So I'm not sure if we would go back to the CGM units as a research sort of purpose. Clinically, they're fine, but research purposes, it was a little disappointing with some of the numbers we saw. But really, what these graphs are showing, so this is, day 2 subtract day 1. So we were kind of interested in seeing, well, was the effect different on day 2 versus the baseline day 1? Or was it different between day 3 and day 1? And like I mentioned, we really didn't see any significant differences. So there was nothing really that exciting about it. Insulin, in this study, we only took insulin at 4 different time points. So insulin was measured pre-exercise, post-exercise, 1 hour post-exercise, and 24 hours later. Okay, so we were just basically interested in a shift in insulin. And again, this is the continuous exercise condition. This is the high-intensity intermittent exercise condition. Continuous exercise condition and high-intensity intermittent exercise condition. And again, we didn't see large changes in insulin. Post-exercise, it did decrease as we'd expect to see. 25 hours later, it was still lower than what we saw at baseline, but not huge sort of shifts. The other thing that we were interested in is IL-6. So why were we interested in IL-6? Well, I was introduced to the immune system a few years back. I was clearly aware of it prior to a few years. But IL-6 in particular is of interest. IL-6 is the original myokine, so the cytokine released from the muscle. Mark Fabreo's group, along with his collaborators over in Scandinavia, identified. And IL-6 is up or increased during exercise. So one of the interesting things around IL-6, it plays a very paradoxical role. So some identify it as increasing insulin sensitivity. A lot of other studies are finding it decreases insulin sensitivity. And in fact, the paper that came out, dual role of IL-6 in regulating insulin sensitivity, is something that's often introduced. And typically anybody who talks about IL-6 and insulin sensitivity talks about its paradoxic sort of role. More recently for me, I'm sure in immunology this has been known for a lot longer, there's a classical pathway and the trans-signaling pathway that's been identified with IL-6. So the classic pathway, IL-6 receptor along with the GP130 subunit attached at the membrane, IL-6 comes in and switches on, multiple pathways. Of relevance to us is the JAK-STAT pathway. Trans-signaling is where the IL-6 receptor is snipped or released by, I believe, its atom, becomes soluble. IL-6 can bond or dock with soluble IL-6 receptor, and any cells now expressing the GP130 subunit, which is ubiquitously expressed throughout the body, can now also upregulate JAK-STAT pathway. The reason why this is important is because the classical pathway is believed to be more the anti-inflammatory pathway, trans-signaling pathway more the pro-inflammatory pathway. And all of a sudden now we have some cells that previously wouldn't be receptive to IL-6 now be receptive to IL-6 because they express GP130 but not the IL-6 receptor. So JAK-STAT in particular down regulates ROS1 phosphorylation. So, and this is via SOX1 and 3. So if you look, insulin attaches, and it basically interferes with the proximal pathway of insulin signaling, so that the signal doesn't come through and the GLUT4 is no longer translocated. Okay, so what did we see with IL-6? Well, I guess the most important aspect was this ratio. So IL-6 to IL-6 receptor, soluble IL-6 receptor pathway. So if we see this ratio go down, we would expect a greater amount of trans-signaling to occur. If we see this ratio go up, we would expect a greater amount of classic signaling pathway to be regulated. And we did seem to see a bit of a shift, okay? And it's appeared to go in the direction that we would expect. But again, this is in an N of 10 in this study. So it is certainly something that we'd have to look at again in a larger cohort. And we are only looking at IL-6, IL-6R, GP130 in separate ELISAs. So this isn't co-precipitation or immunoprecipitation that we're using. So we are just looking at an increase in this, versus GP130. So additional studies could kind of shed a bit more light on that. So I guess just to flip gears a little bit, so what's the training effect? What I've talked about so far is an acute bout, people sitting around the lab for the next few hours. That last study was coming into the lab, doing exercise, and then being released into the wider community, but then coming back on subsequent days. And I do have to mention dietary diaries and things like that were kept and were tried to kind of maintain consistent as much as possible in that study. And it was a randomized controlled study, which is, registered in the clinical trials. So the third study that we're interested in looking at is the training effect. So, so we've done acute exercise. We're actually not seeing much. Well, what about an acute sort of, or training sort of study? What is the training effect? And again, it comes back to this idea that, well, an acute bout of exercise, increases a lot of the hormones, it increases inflammatory markers. Maybe with training, what we see is that those unaccustomed to exercise have a much greater inflammatory response than those individuals who are accustomed to exercise. And maybe it still has something to do with this kind of, the difference in inflammatory sort of markers. So participants for this final study, and again, this is a small study, N of 13, confirmed type 2 diabetes, 51 years of age, relatively good control, not great control. So this is around 7.1 in the old language percent. Typically, we'd like them to be under 7%, but this is also one of the other problems. Individuals who identify with exercise studies who are type 2 diabetes are often in fairly good glucose control. They're fairly aware of, some of these sorts of healthful activities. Slightly obese. I think our cutoff was 27 for BMI. So overweight and obese. Fasting blood glucose is fairly high and fairly low fitness levels. Okay, so what did we do? This is actually a short-term training study. So individuals came in, they exercised every day for 12 days consistently. So day 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. In case anybody wasn't sure how to count up to 12. Once I started along that line, I couldn't stop. But anyway, so training 12 continuous days, 45 minutes, supervised, so individuals came into the lab. Glycemic control, typically HbA1c is used. The problem with HbA1c is that it gives us a fairly good snapshot of the previous 10 weeks. Okay, our training study is obviously only 2 weeks. So we used a marker called fructosamine, which is more indicative of the glucose control over the prior 2 to 3 weeks. So it's more of a short-term marker than what HbA1c is. And then we looked at inflammation, and this we did a bit more of an intensive sort of investigation. I saw this data for the first time yesterday, so I'll present the data, and I'm sure some of you in the room will probably understand it a bit better than I will. But we're particularly interested in the Tregs because they've been implicated in particularly the adipose sort of inflammatory, pro-inflammatory, anti-inflammatory effect on macrophages. Okay, so what did we see? Fructosamine, pre-post, there was a little bit of a difference. It was significant. Was it clinically relevant? Well, probably. But, with fructosamine, there's not a lot of data out there, but really it was a statistically significant shift. It's difficult to kind of tell you. HbA1c, lots of longitudinal studies that are out there, so we can tell you a 1% shift has, particular types of benefits. Fructosamine, the data is not quite out there to kind of demonstrate what sort of a clinically relevant shift this is. But remember, this is 12 days, fairly large variance within the sample. So, we're quite interested or quite impressed that we actually got a little bit of an improvement there. Glucose area under the curve. Well, it went in the right direction, but only actually quite a small effect. Okay, and I realised I was looking at these, but these are actually standard error bars. Okay, so not deviations, and the variance is actually fairly large here. So it is actually a fairly small effect that we saw on the area under the curve for both glucose and insulin. Insulin, you typically see a larger effect. Insulin's more sensitive to the effects of exercise, and I think I mentioned that previously. C-reactive protein, surprisingly, we saw a difference. Most of the larger studies seem to indicate 10 years ago CRP was the big thing, and then the last 5 years none of them seem to see much of a difference. But we seem to see, again, this is only after 2 weeks, so there was a decrease in CRP, which is something that we were quite interested in. And one other thing that was somewhat unique, so we did look at unstimulated and stimulated cytokine populations. This is outside my wheelhouse, but basically what we're interested in doing is we're interested in doing this before the first exercise session and then immediately after the first exercise session. And then we looked at this before the last exercise session on day 12 versus the last exercise session on, immediately after the last exercise session on day 12. Because we were kind of interested not only in the pre versus post, but also with this sort of acute effect, whether or not we see a shift in this, increased concentration of IL-6 immediately after exercise. Okay, that animation didn't work. But basically, I forgot to hide this aspect. But really, we didn't see anything in this, did we? In this, the panels. Sorry, this is Aaron who did the study. And if anybody has questions on the data behind, we'll come back to it. But the reason I presented it all on this sort of page is because we really didn't see a lot in the unstim or the stim sort of conditions. We also looked at some of the cellular markers. So we did some flow cytometry on this, where I sat in my office and wrote different things, and Aaron did the work. We looked at T cells, cytotoxic T cells, and then the Tregs. And then the FOX3, P3 positive Treg cells as well. And really, we're not seeing a big shift. This may have been, was there an interaction effect? Okay, so there's no interaction effects at all in the first pre-post and pre-post the last session. So, again, this could largely have been to do with the large variance in an N of 10. So, in a larger population, we may see something, and this will probably be a subject of, kind of pilot data or preliminary data for a subsequent grant next year. But apologies, this is only just finished, and I've only looked at this in the last week or so. But we really didn't see much in the cellular sort of populations. So, we're a little disappointed with that. We saw improvements. So acutely, we're not really seeing a big shift in glucose, which I guess is no surprise. After training, we're seeing a little bit of a shift, but again, the training was fairly short term. One other thing that's become of interest for us is the idea of timing. We know diet's very important. We know time restriction on food intake seems to have quite beneficial effects. But nobody's really talked much about the timing of exercise. So I guess the second last study I wanted to talk about, and these are a lot quicker, is the effect of training morning versus evening. For years I've always been asked what's the best time to exercise, morning versus evening. And we know with respect to kind of lipid profiles, morning exercise seems to be more beneficial. But with respect to glycemic control, we really don't know. So experimental overview. So this is my other student, Sean, who's just finished this. So basically baseline assessments, we then did 12 weeks of training and then a post-intervention testing. We measured a whole heap of stuff. I'll present the work that he did on HbA1c and glucose response. We used what's called a mixed meal challenge, which is not just a huge load of glucose, the kind of oral glucose tolerance test, but rather a mixed meal which has an increased glucose or carbohydrate intake, but also has fats and proteins, as it's a bit more reflective. So there's some discussion in the literature whether or not an oral glucose tolerance test is the best way to do this, given that it's quite a strong sort of challenge on the system. 75 grams of glucose into the plasma, which currently only has 4 grams of glucose circulating, it's actually quite a large sort of stimulus. So we used a mixed meal challenge here. And I'll talk mainly around the HbA1c and the glucose response. Who were the participants? 40 individuals, 20 did exercise in the morning, 20 did exercise in the evening. We were focused on mainly type 2 diabetics to begin with. As recruitment was slowing down, Sean was starting to panic that he wouldn't finish his PhD within 6 years. So I allowed him to now start recruiting overweight and obese individuals as well. So this is a mix. This is randomized, prior to intake. So we have 20 in here, 10 type 2s, 10 overweight obese, 10 type 2s, 10 overweight obese. And these individuals were randomly allocated to evening, morning, irrespective of preference, which is difficult with exercise studies. Obviously, some people come in with a preference, and unfortunately, sometimes, they don't get their preference and may not adhere necessarily. But such is the life of randomized controlled trials. They're fairly well matched across. There was a difference in age, I noticed, with the PM exercise group being 6 years younger than the AM group, but the type 2 diabetes duration was similar, which is probably the most important outcome here. And anti-diabetic medications were reasonably similar, with metformin being the most prevalent. I wasn't going to show this effect, but I guess one of the things that we did see, one of the reasons why I did want to introduce this was the fact that, often we don't see changes in BMI, okay, and that's because obviously it's comprised of muscle and fat. Here we've got a body fat percent change, and you can see body fat percentage change quite, substantially more than what did BMI, and this is as expected. The exercise protocol was a walking and some resistance training, and you can see some individuals actually increase BMI. Okay, so hence that's just this increase in muscle mass with a decrease in with fat mass. So it is important to kind of look beyond BMI. So what happened with HbA1c? We got decreases. Okay, so it decreased after 12 weeks, 0.27% and 0.25%. When we split this out to the overweight, obese, normal glucose tolerant and type 2 diabetics, we see a difference. Okay, so here we see a much greater shift in the type 2 diabetics. With metformin, after about a year, we'd expect about a 1% shift. So it's not as large, obviously, as what we see with a kind of mono kind of therapy via one sort of medication, but, you know, or oral medication, but it's a fairly substantial effect after only 12 weeks, training just 3 days per week. So, you know, and it was significant. Now, sorry, I've got to emphasize there are no morning versus evening effects. Everything I'll talk about here is just, you know, main effect of time. We didn't see any differences between the AM group or the PM group, which was really our primary hypothesis. But we did see, you know, shifts pre-post training. Fasting glucose, there was quite a large effect on fasting glucose, which we're a little bit surprised at, because usually we would expect a larger effect on the postprandial sort of glucose response. And we saw quite large shifts in glucose and fasting insulin. So again, decreases of 13 and 17 points in type 2 diabetics. Clinically, was it relevant? Well, typically the HbA1c goal is to get people under 7%. Okay, so at the baseline level, 8 individuals in the PM group had a, you know, HbA1c over 7. 10 individuals in the AM group had a HbA1c over 7. Post-exercise, it went from 8 to 6 in the AM group and 10 down to 4 in the PM group. So clinically, it was relatively important. However, they went from about 7.1 to 6.8. So just bear in mind, they shifted into a different category. But, you know, as we demonstrated previously, it's not a massive sort of change in overall units. Postprandial changes are also very important. So if you can imagine, every day our glucose goes up and down, you know, peaks and troughs. When you look at HbA1c, both the fasting glucose contributes as well as the postprandial glucose contributes. Okay, and in fact, postprandial glucose, how high your glucose goes, actually contributes more to HbA1c than what does the fasting in individuals who are kind of in that low range that we utilised in our study. So what did we see there? Well, we saw quite a large change. So zeros from baseline. So we actually saw around a 1.31 unit drop from baseline in the mixed meal challenge. Now this is over this is a 5.5 hour period. 2 meals were presented, and the individual, you know, the drop was actually about 1.31 millimoles, which is actually quite important in this sort of a population. That is the overall, including overweight, obese. Type 2 diabetics, it was 1.45, and those in the overweight, obese category was 1.17. I'll skip a couple of slides because basically this is just you know, demonstrating similar sorts of changes. Okay, so glucose area under the curve. I'll talk about this broadly again. You know, we saw the overall change in glucose area under the curve because we obviously saw that, you know, shift that we obtained postprandially. And again, there was no difference between those who trained in the AM versus the PM. Okay, so the question is timing important? Well, AM was about the same as PM. So exercising pre-12 o'clock versus post-12 o'clock didn't seem to have much of an effect. But one thing to remember is we're only assessing or looking at the effect diurnally, so morning versus evening. And the reality is there's some more recent evidence which seems to suggest the timing relative to meal ingestion is very important. With post-meal being more beneficial on glucose control and exercise in a fasted condition being more important on lipid profile. So we did a systematic review, which I wish we would have completed prior to the initiation of the randomized controlled trial. But, you know, basically what we see, so these are, and again, there's not many studies out there, pre-meal exercise versus control. This favors exercise, favors control. So typically pre-meal seems to indicate, a benefit on, kind of glucose control. Not a huge benefit, but there is, it kind of seems to influence or favor the glucose condition. We didn't run a meta-analysis on this as we planned because the data was so varied, there's so much heterogeneity, it was difficult to add into a sort of a meta-regression or meta-analysis. Post-meal exercise versus control seemed to be a little bit stronger. So the evidence behind post-meal on glucose control seemed to be a bit more favourable. But again, there was no meta-analysis. So the systematic review, as always, insulin, we found similar effects. The effect post-meal seemed to be better than pre-meal. So, how does this relate to the kind of previous study? Well, the question that we have is, those individuals who exercised in the evening, did they now simply push their meal patterns backwards? Or did those individuals eat prior to the morning training sessions? And we literally have a follow-up questionnaire that we're now sending out to our participants asking exactly that. We have food diaries, but we want to ask whether or not they felt they kind of pushed their meals forwards or backwards. And this is the final slide. So exercise training typically improves fasting glucose and HbA1c. Okay, in our study, we see clinically relevant changes. If these studies are done over a 16-week or longer period, often these changes can blow out to around 0.8 to 1%. We saw changes in the vicinity of around 0.4%. The role of the immune system in some of these changes, the immune system is certainly affected by exercise, but we don't really know what its role is within the landscape of glucose control or insulin sensitivity. And really, we need to follow up these studies with larger N. And despite the large volume, there's still much we don't understand, partly because of the large heterogeneity within the data. And we'd love to kind of run clamp studies in the future, but these are not cheap and difficult to do on a PhD budget. Apologies, went a bit longer than I'd planned, but thanks for that.
Thank you very much. Very interesting talk. Thank you. Well, it brings a lot of information about how glucose is regulated through exercise and the timing of exercise. I have a question regarding the effect on the immune system.
I knew you would, Jerome.
So you showed that after the 12-week program, was it?
Yep.
You could see an increase in the regulatory T-cell population from about 0.5% increase.
That was the 2-week program.
Okay.
Yeah.
Even a shorter time.
Even shorter time.
And CD4 T cells and CD8 T cells were unchanged, roughly.
Yes. Yep.
Did you have a look at activation markers on the cells?
Yep.
We ran color panel, so that might be stains and some other stains. But we didn't have enough capacity to have further.
And this is blood, drawn from blood only. So we haven't looked at tissue sort of cell populations.
Yeah, but the fact that there are changes in the regulatory T cell population could be very interesting in some pathology. So, specifically when you look at changes in the cytokine levels. That indicates.
And chemokines was something that we've talked about in the past, but we haven't. We didn't have the capacity to look at the chemokines specifically.
Just before I question to the room, I'm just going to switch on the microphones. We have a better take. If the microphone is close to you, feel free to take it. So since it's recorded, the sound will be much better. Thank you. So questions?
Yes, hi.
Very interesting. In terms of non-exercises relieve some stress levels.
Yes.
Are there any stress markers that you would want to look at?
Yeah, so we've run a couple of separate studies in individuals with clinical depression, because one of the things that I'm often quite interested in is the overweight, obese, type 2 diabetes. So a few things tie these two things together. Inflammation has been identified as kind of progressing, possibly obese to type 2 diabetes. There's a really nice series published in 2005 in Science that kind of talked about that. but we also see kind of clinical depression increase. So typically incidence of clinical depression is around 7% in the population. In overweight obese individuals is around 25%. The problem with that is clinically, individuals who progress to type 2 diabetes with clinical depression are less likely to adhere to medications. So they are the ones who are more likely to have emergency hospital sort of visits. So, and serotonin underlies a lot of this as well. Obviously, it has a role in inflammation, appetite, and depression. So, you know, we're often quite interested in looking at these particular markers. Yeah, so, but again, you know, a lot of them are confusing. Serotonin is one that I've been very interested in, but because of the kind of large family of receptors with serotonin, you know, I don't really know what I'm looking at when I'm looking at an increase in serotonin centrally versus peripherally. Obviously, it doesn't cross the blood-brain barrier. So peripherally, 90% of serotonin is located peripherally and in fact in the gut. You know, in response to a meal, it is released, it then circulates. It has, you know, effects on the muscle. It also has effects on various other areas. So it's, yeah, so mood is something we're very interested in. My colleague has done a fair bit of work on the autonomic nervous system, so doing tilt testing in individuals type 2 diabetes. And there's certainly dysregulation in the kind of autonomic nervous system. Whether or not that precedes or occurs post sort of an increase in glucose, you know, I don't know if people have really looked at that. But yeah, there's a whole host of things, and it becomes problematic. My background is more metabolism. Endocrinology is complicated, but the immune system, I'm starting to find out, is worse. So I'm starting to kind of step away slowly from that aspect. But it's a, yeah, so, but yeah, now to answer your question, there are a whole host of sort of different areas that we're interested in particular with weight, depression, as well as appetite. That we're kind of looking at these markers. And we're also interested in GLP-1 and a few of the sort of small peptides released from the stomach that not only stimulate appetite, but also are incretins, so stimulate the release of insulin. So we've looked at GLP-1 and GIP as the two primary incretins. And the problem with some of these markers, glucagon especially, because in the past, you know, the role of glucagon on the liver has been underestimated, because the reality is the pancreas sits behind the liver. So as a researcher, we're sampling peripheral blood. The pancreas is released, first passes through the liver. So the problem is, you know, research-wise, the liver's a pain in the bum. Physiologically, it's in the perfect position. So the reality is the rise in glucagon that you often see with exercise is we're not seeing much and it's not that interesting, but in using a dog model, they're seeing huge increases in glucagon, and it really is the one that influences the release of glucose from the liver. And the dog is used as a model because you can access the portal vein very easily in the dog model, whereas, you know, in humans it's very complicated. But yeah, so there are a range of things that I'm interested in looking at. Yeah, it's always costly as well. Yeah.
In terms of, you mostly looked at type 2 diabetes.
Yeah.
What happens if you looked at healthy controls in sedentary versus exercise?
That's a good question. We often get that one from reviewers as well. Yeah, look, we haven't done this in just sedentary individuals who are unaccustomed to exercise. It's one that we've had in the past, and I've got to be honest, I actually don't think there's lots out there. Exercise immunology has 15 years ago was done by exercise physiologists doing some immunology, so dabbling. Now what we're seeing is proper immunology work coming together with exercise and doing more, rather than just descriptive sort of immunology, are doing much more sort of sophisticated, looking at, you know, migratory or, you know, chemokines, and, you know, actually looking at tissues specifically. And I suspect, especially my collaborator at the moment, is doing some work with Professor Jensen in the US, and they're looking at clamps for sort of lipid clamps. And they're very interested. Mike Jensen does a lot of work in adipose tissue, just a subcutaneous sort of adipose tissue, and they're very interested in looking at, you know, what's actually happening in the differentiation of some of these T cells and the Tregs, you know, in the adipose tissue. Because there's very nice work in animal models with the crowning that we see with some of these sort of macrophages, but we haven't done a lot in the human work yet. So sedentary, normal or normal glucose tolerant, there's not much work done, and we may have to take a step backwards before we take a step forward.
UWA had an interesting study about 8 years ago that they found like a 7-Eleven recipe. Actually, you burn more calories and build better muscles faster, enhance the metabolic rate quicker by doing 7 seconds, resting 11 seconds, running, and you just keep doing that for several minutes. I wonder if you did something like that, if you see some improvement in
Yeah, I'm actually not familiar with that one. So they did 7 seconds on...
So the subjects on a treadmill, and they had to run for 7 seconds. Run for 11 seconds, and then go into walking mode for 7. And it's that initial start, like
Yep.
you use way more energy to start, and it's a bigger hit on the muscles to repair, grow.
Yeah. It's a good question. I'll have a look, because most of the work we do is with collaborators at UWA. But it's intensity is key. It really is. So when you think about it, it makes sense, because even some of the work that Michael Mosley did with some of the stuff that Michael Mosley presented all those years back on the sprint, because the reality is glycogen is a fuel source the muscle preserves for fight or flight situations. So in everyday life, rarely do we utilise any of our glycogen. So if I take a muscle biopsy, and if any of you want to volunteer, just meet me up in my office, muscle biopsy, measure glycogen, 3 days later, irrespective of diet, glycogen, even if I keep you on a fairly low carb diet, muscle glycogen's at about 80%, even if I keep you on a kind of fat diet. So the muscle preserves glycogen at all costs for flight or fight response. We are animals. We used to run away from dinosaurs. I know we didn't. So we use that. So the reality is we need to increase intensity to break the glycogen. And in fact, we can utilise glycogen. My honours study was looking at glycogen utilisation, start with 100 units, 3-minute sprint, we bring that down to about 45 units. So we can rapidly deplete muscle glycogen. The body strongly then has multiple pathways where it can replenish that muscle glycogen, because it needs to be able to replenish muscle glycogen very rapidly. But the intensity is key, because if you don't deplete the muscle glycogen, then why would the muscle become sensitive to taking in more glucose? And the reality is, insulin resistance is a perfectly normal is a perfectly normal response to an abnormal situation. The muscle takes glucose in. The problem with muscle is glucose comes in, hexokinase has a really low Km. As soon as glucose comes in, it is phosphorylated to glucose 6-phosphate. It has 3 options: pentose phosphate pathway, not very active in muscle, glycogen synthesis, or the utilisation in the glycolytic pathway. The problem is, we don't always need ATP. Glycogen builds on primers of glycogenins, so we can fill those stores up. Pentose phosphate pathway is not very active. The muscle doesn't want glucose, so it becomes resistant if we have high levels of glucose. Making it more resistant is just going to cause downstream problems. So, in order to kind of deplete the glycogen, the muscle becomes more receptive to more glucose coming in. So, so when you kind of think about this progression from insulin resistance to type 2, it's actually a fairly normal sort of response. It's a very clever response that the muscle kind of sets up. So if we don't bring this glycogen level down, then it's going to cause problems. So it is absolutely intensity. Does it have to be 5 seconds, 10 seconds? And I know that's not your question, but there's always this debate on how intense does it have to be. But it does need to be intense to act or to kind of bring the glycogen down. As I mentioned before, different fibre types have different sensitivities as well. So in order to activate the type 2 fibres, it needs to be fairly intense sort of activity. So intensity is very important. Most of these studies are fairly low intensity. Walking, we may not have seen a depletion of glycogen. In fact, we would have seen a very low mobilisation of glycogen at this sort of level of exercise. So higher intensity exercise is good. Ethics committees often don't like us to do high intensity in clinical populations, though. Sorry, what was that?
Is it correct that basic exercise, and according to just all your data, does nothing?
No, exercise actually does a fair bit. So what, because one thing just to remember is we're looking at one marker, glucose. And what we're seeing is quite a large difference. HbA1c decrease of around 0.4, 0.5% is actually pretty big. And like I said, metformin is around 1%. Add metformin with, one of the other sorts of drugs, TZDs or whatever it might be, sulfonylurea, you may see 1.6, 1.7 sort of changes. But exercise, now remember, this is 3 bouts, 45 minutes in 1 week. So, we're doing 160 minutes of something over the course of an entire week for 12 weeks, and we see a significant difference. One thing we've got to remember is type 2 diabetes doesn't occur overnight. So it's a clinical condition that progresses over a long period of time. And often we think, well, I don't understand, exercise didn't do anything. 4 weeks, here we go, I tried really hard. Didn't alter diet. This is one other thing. We had no restriction on diet. And some people compensate fairly, they increase the dietary intake. So addition of diet and exercise has a greater effect. So we would see similar sorts of effects as what you would expect with metformin, etc. If we do a dietary plus exercise intervention in 12 to 16 weeks, take that longer, we will get a larger effect. And you can certainly reverse the type 2 diabetes, bring someone into a normal glucose tolerant range. So ours are probably smaller scale. Remembering these are just, PhD projects unfunded. $6,000 was the budget for some of these, which disappears with just the kind of catheters alone. But we do certainly see differences. Clinically, the idea is, you don't see a big difference, but the reality is you see a fairly large difference. Acutely, you don't see much, but clinically you do.
It's getting a bit late today, so if you have more questions, feel free to come and ask directly to Tim.
Thank you, great. And you send me